new concept and wonderful initiative from our younger faculty colleagues from CAS and Dr. Sultan Habibullah coordinated this effort that we are going to discuss CRISPR-Cas applications. I have discussed with high officials in Islamabad, and the government is very keen on creating capacity on applications of CRISPR-Cas for genetic editing and more specifically in agricultural fields, both crops and livestock. So today's session would also act as a kind of baseline discussion of what we call as in-house feasibility session. This is what is required for any planning commission project. So I will ask Dr. Sultan Habibullah to take good notes. I'll try to take my notes as well. But end of the day, we would like to get a consent from the participants in terms of supporting a project proposal to be submitted to Government of Pakistan for funding to secure a long-term commitment in terms of collaboration with our overseas collaborators, China and elsewhere. We are trying to get some collaborators from Germany and the United States of America. We have young faculty members who have already had a good handle of working with this technology. They are also logged in, and I will ask them to participate in the discussion and create a network that our project proposal gets strong commitments from partners from within the country as well as from overseas. So with these few words, I welcome our keynote speakers who have very kindly agreed to share their valuable time with us, and we stand benefited because of their presence, and also we hope to learn from their experience as well as find out what are the new avenues that we might be asking through a project proposal for funding from federal government of Pakistan. And also the provincial government is keen. Today we had a visit with Dr. Abad Mahmood, who is Chief Executive Officer of Punjab Agriculture Research Board. And ARB is also equally interested in developing capacity and investing into problem-solving applications of CRISPR-Cas. Okay, so with these few remarks, Sultan, please, you can take over and invite the speakers in the sequence you want. Over to you, Sultan.
Thank you very much, sir. So, Dr. Holger is our first speaker. He is well-known scientist in plant biology, especially double-strand DNA breaks. So I would request Dr. Holger to start his presentation.
Well, thank you so much for inviting me to this very interesting meeting. And I will now try to, in principle, cover a little bit also the historic perspective, how things evolved over the years, and how we are, where we are now in this moment. So that's why the title is Inducing Double-Strand Breaks in Plants from Gene Editing to Chromosome Engineering. And I will cover three different aspects. I think gene editing, this is where most people are interested in. I will shortly give you a historic overview, why and how long this has been done already. And I will also cover very shortly politics in Europe, because this is something that's unfortunately not so nice as we hoped it would be. Then I will cover gene targeting. This is a special way of editing by using homologous recombination to change genomes in a pre-planned way. And in the end, I will tell you a little bit about our most recent results, which make me quite enthusiastic that we are not only able to edit genes, but by now we are also able to edit chromosomes with this technology. So let's start with gene editing from history to politics. And I have to start with simple plant breeding. And of course, most of you are well aware of what is breeding all about, but I want to make it very clear. And I want to show you here in this respective slide. In the middle, you see a plant. This is a brassica plant. And here you see all of these different kinds of monsters that breeders, vegetables that breeders made out of this brassica plant. So this brassica plant had the genetic potential to get to Brussels sprout or to get to broccoli or to get to kohlrabi, cabbage. But the trouble by now is that if you have all these different kinds of crops, the genetic diversity is gone. So nobody is able to transform Brussels sprouts again into kale, for example. So in principle, this is a very general problem we have with crops, that genetic variability is gone, and we have to somehow to get new traits introduce genetic variability again. And in principle, a very brutal method, a method that has been applied, as you will soon see, much longer than you might think, is by inducing double-strand breaks into genetic information. And by inducing a double-strand break, what you do is obviously, in many cases, you harm the genetic information. It has to be repaired, and this repair leads, as is shown here by end joining, to a change. And if this break is within an open reading frame, the open reading frame might be destroyed. So in principle, you might argue, well, such a technology might not really be beneficial to develop new traits. But if you think about agricultural traits, some of the traits are in principle also killing genes. So think about grains, for example. Cereals, in nature, they have to have very long, thin stems to survive. But if it comes to a field, you need short stem cereals, because if a storm comes, all other cereals will be broken by the storm, but not short stem cereals. And so if you, in principle, are able to induce such a mutation, you are producing a beneficial trait for agriculture. And this technology was indeed applied for a very long time. As most of you, I hope, know, by just irradiating plants with radioactivity, you are able to induce multiple breaks in the genome at the same time. And this technology, sometimes some people also use chemicals for this, is used since more than 70 years for breeding. And it's in principle a very successful technology. The trouble with this technology is obviously that you induce many different breaks at the same time. And this means that you induce many different mutations at the same time. Most of the mutations are not helping. Most of the mutations kill your plants, but in principle, once in a while, you get a good trait like the short stemness, which I told you before. But the price is that you have to cross it out, you have to cross it back, and this takes a long time. But nevertheless, this is very successful. I don't know whether some of you had a grapefruit this morning. So if it's seamless, grapefruit ruby red was produced with this technology. Maybe some of you had a beer this morning or this evening. I don't know. It's later in Pakistan than here in Germany. People, at least here in Germany, drink beer. So also short-stem barley in Europe is produced with this kind of technology. So we have 3,000 cultivars worldwide that are produced this way. And in principle, already in the last millennium, it became clear that in principle, nature has much better means to induce breaks. And obviously, there are a lot of different processes during the lifetime of organisms where you have to recombine or make new combination of genetic informations. And whenever you have new combinations of genetic information, you have to cut DNA first. So there are a lot of enzymes in nature that are able to induce specific breaks. And of course, to apply these technologies to plants or to higher organisms, higher eukaryotes, you would have need enzymes that are specific enough to induce just one single double-strand break in the genome. And we went in this direction, and indeed in 1993, a very long time ago, we were able to apply the first enzyme IC1 at this time, that we could induce a single break in a plant genome. So the recognition site of this enzyme, you could only cut one site with this enzyme in a genome which you pre-inserted before. But in principle, now we were really able to ask the question, if we induce a break in a specific position, what can we achieve with this? So in principle, since then, me and my group was concentrating on these questions. What kind of different changes can you achieve by inducing an enzymatic double-strand break in genomes? And what we found out, and this will be interesting in the further in my talk, is that the main pathway is non-homologous end joining if breaks are repaired. So the ends are formed together, but this non-homologous end joining are indeed two different mechanisms. The classical mechanism, often one or two nucleotides are lost and is mutagenic, so open reading frames might be destroyed this way, or you have this alternative non-homologous end joining. There a few microhomologies are involved, and when they are rejoined, then there is a deletion occurring, and this is a highly mutagenic pathway. And as you see here, we know these pathways since more than 20 years. And interestingly, also at this time, we also found that end joining or inducing double-strand breaks using end joining, you can also integrate DNA. So what we found, and we were very astonished at this time, that into these breaks, if we go in with a tDNA for transformation into these plants, we can also found it integrated in the double-strand break. And by now, everybody knows, or this is generally accepted, that double-strand break repair is a mean how also foreign DNA, tDNAs, for example, are integrated into plant genomes. But at this time, this was, of course, surprising, but it gave us the chance to learn much more what we can achieve using double-strand breaks. And then we got this historic development over the years where a lot of people were trying to improve the technology to develop new kinds of enzymes that really direct you to different parts of the genome, that you can really go for the gene of interest you really want to change in the genome. There was the zinc fingers, there was a transcription activator nucleases. All of these systems worked more or less well. However, it took a long time to get these enzymes, and they were very expensive. Zinc fingers, we had to wait, for example, for a year, and it cost us $25,000 to get a single one. And all of these dramatically changed in 2012. In 2012, the CRISPR-Cas system was discovered by Chapoutier and Doudna. And as you all know, I'm sure you all are aware of the fact that this year, finally, both of them were awarded with the Nobel Prize. I think this is really, really one of the major transformations in biology we have seen over the years. And this new thing, the exciting thing with this new technology is in principle that now you have a protein, but this protein is assisted by an RNA, by a guide RNA. And this guide RNA is very simple to construct. And with this guide RNA, you are able to address in principle any kind of sequence in the genome. And this gives us the opportunity in principle to get these enzymes in a day and for $20. So this is a big difference. And on top of that, these CRISPR-Cas nucleases are much more efficient in cutting DNA than all the previous enzymes evolved. And on surplus of all of this, you can also use several guides, as you see here, the guide, at the same time, and you can even induce more than one break at a time. So all of this led to the consequence that really an explosion, a revolution took place in biology. Also, the basic mechanisms and enzymes were developed long before CRISPR-Cas was discovered. And we know for 30 years what we are indeed doing if we're inducing double-strand breaks in eukaryotes. So in principle, when we started to work with CRISPR-Cas in 2012, there was a problem that was till then not solved. This was the problem that nobody was really able to induce mutations over generations in Arabidopsis. And this is Frieda Fauser, a PhD student in my lab, who was really able to solve the problem. And in principle, as you might know, if you are plant experts, for Arabidopsis, you need this XL transformation protocol, and everybody was using the 35S promoter, and then these events were not transferable to the T2 generation, but we used the UBI promoter to express Cas9, and here you see the Cas9 open reading frame, here you see the RNA that is expressed, and using this technology, we were then really able to induce mutations in Arabidopsis. And in principle, at this time, we were, of course, highly happy to get mutation efficiencies in the next generation, about 15%, which was much better than anything else that could be achieved with zinc fingers or TALEN sequences. But in principle, as my group is interested also to find events that do not occur very often, it's important to get to enzymes that are really cutting extremely efficient. And therefore, we not only tested Pyogenes, the classical Cas9 nuclease, but we also tested other CRISPR-Cas nucleases. And we found out that the closely related CRISPR-Cas9 from aureus, and this was work done by my PhD student, Janet Steinert, had tremendous efficient cutting efficiencies in Arabidopsis. I show it here. You see, for this specific sequence, we got over 90%. Of course, we have to be always careful. All enzymes have specific sites in the genome where they cut better and other sites where they're not as efficient. And you always have to be careful. But overall, we are very happy with the aureus Cas9, and we are using it for many different things by now. And you might also know that besides Cas9, a few years ago, also a second enzyme class was discovered and developed also as to engineer genomes. This was the Cas12a class. The problem with Cas12a is that for plants, these enzymes are not very efficient because most of these enzymes are from bacteria that are pathogens of humans. So these enzymes are optimized for 37 degrees. And it might be very difficult to use them at 22 degrees where plants are cultured. And so we developed an enzyme, LB-Cas12a. We optimized it for temperature tolerance. So it was known that certain kinds of mutations can enhance the efficiency of these enzymes. LB-Cas12a was an enzyme that is working best in plants. And so we optimized this enzyme recently. And by introducing, testing a certain number of mutations, we found out by a single mutation, we are able to indeed enhance the efficiencies quite drastically, as you see here. So this is the original enzyme. Here, the blue one, and this is the temperature tolerant enzyme, which we got. And you see all over all these things. I don't want to go into much detail. We can enhance the efficiency 3 to 4 times. And if it comes to events that are not occurring very often, like gene targeting events, you will see later on, it's very important to really work with the most efficient enzymes. And let me state that if anybody of you, anybody of the audience is interested in these kinds of enzymes, I'm very happy to supply you all with these enzymes. If you are starting projects, also in different crops, it might be very interesting for you to test whether these enzymes are not much more efficient than the classical Cas9 enzyme. So if you write me an email, we are happy to send you the material. So in principle, I told you I shortly want to mention also the situation in Europe. So editing is working quite well. A lot of traits have been produced. I'm sure that Chaisha Gao will also tell you more about this and all the different traits she achieved in different crop plants. But in principle, if it comes to risk assessment and what it means also to have these plants in the field, you have to see that double-strand break repairs are a natural process. molecular scissors are known and are used since 25 years. So we have a lot of experience with this. These methods are extremely quick. They are precise. They are hardly, if at all, offsites. And there is no way to discriminate a plant that is induced by a CRISPR-Cas from a natural plant. So all these mutations are natural, identical. However, in Europe, we have this crazy situation that the European Court of Justice ruled in 2018 that these plants have to be treated like GMOs. On the other side, classically mutagenized plants, as I mentioned before, have a long safety record and they don't have to be regulated. And this, of course, means that in Europe, we are at the moment not able to use CRISPR-Cas plants in the fields. Also, we are able to use these classically mutagenized plants in the fields. And this brings us to the following situation. If you think about how many mutations you induce with classical mutagenesis, even in the field, you have plants. If it comes, for example, to barley, you have plants in the field that have 10,000 offsite mutations, and this is fine for everybody in Europe. If you take two natural barley plants from the same field and compare the genomic sequences, you will realize that these genomic sequences are not identical, but you have a natural mutation rate that is almost 100 per plant. So in principle, also two natural plants in the same field differed by 100 mutations. And then you think about our CRISPR-Cas technology. We induce a single mutation. So in principle, we are below the noise of mutation. So we are really better. I always make this very provocative statement. We are better than nature by now using CRISPR-Cas. And these systems are in principle at the moment banned from growing in Europe. And this is for us European scientists, of course, a situation we cannot accept. And I talked to the German ministers of agriculture. I talked to the German minister of science. We talked to the European representatives of the European Parliament. I'm involved in many organizations, the German National Foundation and the European academies. And a lot of European scientists at the moment try to lobby for this new technology in the hopes that also in Europe it will be possible to grow these kinds of plants. Okay, but coming back to basic science, because this is more rewarding, at least for me as a scientist, gene targeting. Gene targeting is a technology where you use homologous recombination to introduce changes in a way you want to have them in the genome. And we could show this already all these years ago in 1996 for the first time in plants, that if you induce a double-strand break and you bring in a transgene, a tDNA with homology, you can introduce at this specific position in the genome this kind of change. The good thing was that before double-strand breaks were used for induction, you had to transform 100,000 of tDNAs to get one integration event in a specific site. Inducing a break, it was 1 out of 100. The bad thing is that we are still struggling since 20 years that we get a much higher frequencies with homologous recombination. And there's a lot of paper published. I will also tell you a little bit about our efforts, but still the frequencies of homologous recombination of gene targeting are much lower in plants than for editing. We developed a technology some years ago, which we call implanter gene targeting. And this technology is based on the fact to avoid many transformation events. So if you have to have hundreds of transformations to get one targeting event, you might argue that this takes a lot of time, especially in plants that are hard to transform. And so we said, okay, why shouldn't the plant do the work? And so we integrated the vector beforehand in the genome together with the nuclease, and then we activate the vector by cutting it out in all cells of the plant, and at the same time inducing a break in the target, and then go for the next generation for the seeds to find the correct events. So I show you, we cut out the donor reacts via homology in the regions with the target, and then it's repaired. You can segregate the donor away, and you have this change. And we set up this technology in Arabidopsis already before the CRISPR-Cas revolution. And then we adopted it using a natural herbicide resistance gene, the ALS locus. So you can, in principle, select for this if you induce a point mutation into the ALS gene. This was work done by my PhD student, Felix Wolter. And indeed, I can show you some results we obtained. So in principle, what we found is that there's quite a drastic variation if it comes to different lines. So you integrate this into the genome, and depending on the integration locus, you get very different recombination frequencies. So we have some lines where we have very high targeting frequency, high, for us at least, targeting frequency of 6 or 4%. Many lines of 1 or 2%, and we got this higher percentage as we used an Excel-specific promoter, as it guarantees that this thing is heritable in the next generation. You see here the UBI promoter, so the frequencies were much lower. So in principle, as I mentioned before, we also were interested not only in Cas9, we are using, of course, aureus Cas9 because this enzyme was working better in our hands than LB Cas9, but we also tried to work with LB Cas9. And there is this line of argument that because here you see with the normal Cas9 enzymes, there is a cut is within the seed sequence of the guide. And so if there is any mutation introduced, this cannot be cut anymore. But it's different with Cas12. And so the idea was maybe we can cut several times with this enzyme till we get a homologous recombination reaction, and that's why it might be more efficient. And so we performed a row of experiments. And here we also checked end joining. So we first performed these experiments with the original LB Cas12a, and we found that it's not cutting as efficient as the aureus Cas9, but nevertheless, if it comes to homologous recombination, the colors are not changed. So the blue color is better than the red color you see here. The rates are a bit higher. Also, we have only about half of the cutting efficiency. So this was very rewarding for us because it indicated that due to the nature of the enzyme, this enzyme might be better for gene targeting reactions. And then we performed the experiment with our improved LB Cas12a just recently. This was work done by my PhD student Laura Merker. And here you see the blue. If you compare the blue with the green, we had much higher frequencies with the LB Cas12 that was tempera optimized. So this is really the best enzyme, at least we have in our hands at the moment, for inducing mutations, but also for inducing gene targeting implants. And as I told you before, if anybody is interested, I'm happy to supply you all with this enzyme. Chromosome engineering. This is my last thing, and this is the newest results I want to present you. And it's also something which we only started recently. And the question is, of course, why chromosome engineering at all? Does this make sense? Well, of course, it's obvious that you might get rid of sequences by inducing a deletion. So you induce just breaks, and if the thing is deleted, we could show this already a very long time ago, still with IC1, that this works very well in plants. But there are two very other interesting things you might do with two breaks. You could also get an inversion if the fragment is integrated in the wrong way around, or you might also get translocations if you introduce two breaks on two different chromosomes. And both of these things are in principle interesting for breeding, because in breeding, you need to break genetic linkages. And if genes are close on the same chromosome, you're not able to do it. So for example, if you have a gene that is responsible for a bitter fruit taste, and this red one, and the black one is responsible for virus resistance, you of course want to get the virus resistance, but you don't want to get the bitter fruit taste. And by inducing breaks between these two genes and by exchanging this part of the chromosome with another chromosome arm, you should be able to achieve these kinds of things. And also inversions are very interesting, because inversions during evolution, during genome evolution, also in crop plants, occur regularly. So you always have these problems that surprisingly you cannot transfer certain traits from a wild relative to a cultivated plant. And it turns out that also the sequences are there, they are inverted, and there is no exchange, possible genetic exchange. So why don't we revert the sequence to get exchange back? And I show you one example from Arabidopsis, because this is our model system. Indeed, in Arabidopsis, you have about half of the cultivars have an inversion, and this inversion is in principle responsible that is in this specific region of the chromosome, you don't get any genetic exchange anymore. And we thought, of course, is it possible that we can induce inversions in the genome? And we started this, and of course, because we are decent people, we didn't start with this 1.2 megabase inversion. We just started with 2 kb. This is work done by Carla Schmidt. And we just analyzed, are we able to get inversions if we induce the 2 breaks? This was all done with Aureus. And indeed, by digital PCR, this was the first line of evidence we could show, yes, we get about 0.5 to 2% of the cases where we induce this break, we get inversions. We get, of course, much more deletions, which we also analyzed. But so it makes sense to go for this. And we also analyzed the patterns, the rejoining patterns. And to our big surprise, we found that most of these rejoined ends are completely conserved. There was not a single nucleotide lost or gained. And in the first moment, you don't expect this, because if you think about editing, there's always mutations. But apparently, because the guide is not able to recognize these new joints anymore, there is no cutting possible anymore. And this indicates that most probably also under normal conditions, if you use such an enzyme, there are multiple rounds of cutting till you induce a mutation, and then it's stabilized. And because we are stabilizing the junctions, because these guides are not able to recognize them anymore, if there is inversion, we see now the effect that they are perfectly rejoined. And of course, we thought, okay, interesting. A perfect rejoining, this is a classical mechanism of non-homologous end joining, as I showed you before, and I show you here again. And there is one protein that is very important. This is the Q heterodimer that keeps the ends safe if a break is made. And so we thought, okay, if we only have perfect events, we take this enzyme out, and then most probably we don't get translocations at all anymore, because all the junctions were telling us that these proteins are involved in junctions formation. And we made the experiments, and we got a big heart attack, because we saw that there is more if we knock out Q. So we have about double amount. So it's a mean to enhance these translocation frequencies, not to reduce them. And we were in principle, of course, surprised. So the alternative pathway apparently is also working well. And the explanation for this is that in principle, this Q heterodimer is not only responsible to conserve these 2 ends, but it's keeping also the 2 ends completely together. So nature has an interest that the right ends are always joined together. But if you get rid of Q, then the ends are free, and there is no control anymore which ends are joined, and that's why we get more. So it was surprising, but now we can explain this, and we can use it to a certain extent also to get more translocations. Also, it might be that some junctions, because you get bigger deletions, are then too imprecise for biotechnological applications. It always depends what you want to do. We used our Excel-specific expression like we used for targeting, and then we found with the short inversions that we got quite reasonable frequencies depending on the lines we screened. Some of the lines didn't show anything, but some lines showed 5 to 10% inversion frequencies. It all depends on the integration of the transgene. And then we asked the question, then we are really, we had enough, we convinced ourselves that we had to go for the price. Let's inverse the 1.2 megabase knob inversion in Arabidopsis. And we performed the experiments. This was also done by Carla Schmidt. And indeed, we were surprised that we got a row of events with this approach. You have to screen for them, and most of them had perfect junctions. So we really inversed 1.2 megabase in the genome, and we didn't lose a single nucleotide. And Paul Franz, a colleague from the University of Amsterdam, who was the one who really discovered this inversion or characterized this inversion in detail, helped us then for the cytogenical analysis. So using bugs, you see here in wild type, you mark this region as red. Sorry, I have to go back. I cannot wait a second. Red, green, red. And the inversion should be red, red, green. And here you see this is a wild type, red, green, red. And here you see the inversion, red, red, green. So it's perfect. I mean, we really were able to inverse this and to show this on the chromosomal level. And then we went for mitotic recombinations. Then we crossed it to the other cultivar, which didn't have this inversion. And then for the first time, we really could induce meiotic recombination in this region between these two different ecotypes, cultivars, where no recombination has been taking place for the thousands of years since the inversion was produced. And this clearly shows us that such a technology, and this was done in cooperation with my good friend Andreas Huben from the IPK Gattersleben, that such inversions can be reverted now also in crop plants, I think. So this is a very important add on our toolbox. And the other thing, as I told you, was translocations. I want to make this very short because my time, I think, is also running out. So this was work done by Natalia Beijing, and she tried to exchange chromosome arms of chromosome 1 and 2 of Arabidopsis. Here we also saw in our preliminary experiments with the Q mutations, we get more events. In principle, the number of events with digital PCR was very low, so we really had to screen a lot of plants to find these events. Screening over several 50 or 100 primary transformants. We went to the T2 and then went to single plants. But also here we got as well from wild type as well from the Q mutant, where we got more. We got a lot of translocations. We analyzed the junctions, and again, in many cases, we didn't lose a single nucleotide. And then, together with my friend Andreas Huben, we made the analysis, the cytological analysis. And as you see here, this is wild type, red and green. And in the mutant, you have the combination. So this is part of the green chromosome combined with the red chromosome. So this worked, and we said, okay, maybe let's try another combination between chromosome 1 and 5. And again, we didn't go for so many events at this time. We just wanted to show the principle is working also for other chromosomes. It worked for other chromosomes. We got it. And here is also, if you see this, I think this is really, really nice. Here you see the green chromosome. Here you see the red chromosome. And here we newly combined the red and the green chromosome. So in principle, we are now able to also restructure chromosomes using the CRISPR-Cas technology. And this gives us, of course, a lot of new avenues we could go. In crops, we can try to break and fix linkages by restructuring chromosomes. But of course, for basic science, it's also very important. We can try to restructure chromosome and genome evolution, because a lot of translocation occurred during genome evolution. Of course, we can use this technology to construct novel synthetic plant chromosomes. And in the end, if you think about that, it shouldn't be too difficult also to construct new plant species by just changing the chromosomal set of a specific plant. And of course, we are going in this direction now with our model Arabidopsis to see how far we can take the genetics with this kind of technology. And with this, I'm at the end, and I want to thank you for your attention. I hope I didn't bore you too much with our results. And I also, of course, want to thank my co-workers, and very important is also for me to thank the European Research Council that over the years funded our research now for almost 10 years with 2 consecutive advanced grants, which really makes a difference and enabled us really to address all these questions, to achieve also all these different interesting results with chromosome engineering. And with this, I thank you for your attention.
Thank you very much, Professor Holger Pochta. You have really set the stage, and we have learned so many new things during your short 40 minutes presentation. A lot of students actually are logged in, and they seems to be excited about the possibilities. We are also grateful to you for having made offers that you are willing to share the materials and all kind of help. We will also probably look for the opportunities to send some trainees to your lab and see if we can benefit from your.
Yeah, at the moment it's of course difficult because of the CRISPR-Cas times, but in future, I hope after if the world turns normal again, this is of course an option.
Thank you very much. Okay, Sultan, who is next?
Right. So our next speaker is Professor Gao from China, Chinese Academy of Sciences. Unfortunately, she is not available due to flight change, but she has shared us with a recorded video. So, Dr. Zulkarnain, can you please initiate the video?
Hi, good evening. So, due to some unavoidable circumstances, Professor Gao couldn't make it to join this session, but she has sent her video lecture for this session. So on her behalf, I'm going to play a lecture, and I hope it would give more insight into the CRISPR-Cas technology and its applications.
Good afternoon, everyone. First, I would like to thank FTech for inviting me and giving me the opportunity to talk about our research work in applications of base editing in crop improvement. I'm Chai Gao, and I work at Institute of Genetics and Developmental Biology of Chinese Academy of Sciences. I'm very sorry I had to make this recorded video in advance. As we know, CRISPR arrays in bacteria serve as an immune memory and defense mechanism. Bacteria programs it to target and cut viral DNA. So viral DNA is destroyed. This viral immunity system has been repurposed for genome editing in plants and animals. So CRISPR can be programmed to recognize particular DNA sequences of plants and animals and make a double-strand break in DNA. So we can trigger cells with programmed double-strand breaks by either the destruction of a gene or incorporation of some new genetic information. So precise genome modifications are achieved by harnessing DNA double-strand break repair pathway. In 2012, the groups of Charpentier, Dogna, and the 60s showed that purified Cas9 can be guided by CRISPR RNAs to cut targeted DNA in vitro. They published their work in Science and PNAS. In 2013, it was half a year later, a pair of studies from Feng Zhang and George Church labs simultaneously showed that CRISPR system can be or can perform genome editing in mammalian cells. So early this October, the Royal Swedish Academy of Sciences has decided to award the Nobel Prize in Chemistry 2012 to Charpentier and Dogna for the development of a method for genome editing. In 2013, the CRISPR was established for use in rice and wheat. So since then, CRISPR has been used by thousands of labs for genome editing applications in many plants and animals. So this technology has become possible to edit a great variety of plant species. Application of the technology is becoming more attractive for agronomical purposes. Crop improvement requires the constant creation and use of new allelic variants. So conventional breeding, such as crossbreeding, can only introduce traits already available in the parental genomes, and it takes also several years to introduce desirable alleles by backcrossing. Mutation breeding has expanded genetic variation by introducing random mutations using chemical or physical treatment. However, mutagenesis is random and non-specific, and generating and screening large number of mutants is very challenging. Transgenic breeding can introduce design traits through the transfer of foreign genes into elite background varieties. Foreign DNA is randomly inserted into plant genome. So the commercialization of GMOs is limited by long and costly regulatory regulation. Genome editing can accelerate plant breeding by allowing the introduction of precise and predictable modifications directly in an elite background. Genome editing using Cas9 RNP has been achieved for lettuce, wheat, maize, and potato. This should make new plant breeding technologies more acceptable for worldwide use. Today, I will focus my talk on basic editing. Beyond double-strand break-mediated genome editing, base editing systems that can induce specific base changes efficiently. The systems do not depend on double-strand break and the donor DNA. So there are two base editors. So Davies group developed two types of base editors: cytosine base editor and adenine base editor. So we call it CBE and ABE. So in CBE, it consists of cytidine deaminase dried FBAC1 fused with a Cas9 linkage and a uracil glycosylase inhibitor. So cytidine deaminase first converts the C in DNA to U, and the U is then replaced by T during replication. 1 year later, in 2017, the same group developed adenine base editors. So these base editors mediate the conversion of A to G in genomic DNA. So very similarly, adenine deaminases first convert A into I, and the I is then replaced by G during DNA repair and replication. So then we know the most widely used cytidine deaminase is right FBAC1. So in my lab, we first tested whether FBAC1 based CBE could edit the coding sequence of BFP to produce this GFP activity. So then this requires a changing codon H66 from CEC to TEC. Therefore, we designed a GetRNA BFP with the design C as a position 4 here and C. Then we introduced all this plasmid into rice and wheat protoplasts by PET-mediated transformation. So this yielded here 6.8% of rice cells with GFP expression, while substituting Cas9 yielded only 0.3% GFP positive rice cells. So we next edited endogenous genes. We designed a guide RNA for each of 3 rice genes. 3 different guide RNAs for one wheat LOX2 gene and one guide RNA for maize CNH3. As control, we use wild-type Cas9 to produce indel. So we call the system as PBE, plant-based editor. So PBE induced the C2T conversion in all 7 target sites with deamination windows spanning from position 3 to 9 of the protospacer. And the frequency of single C2T substitution was 0.39 to 7%. And the highest efficiency was at position 7. We also observed a lot simultaneous editing of 2 or 3 Cs. To regenerate base editing mutant plants, we targeted a rice CDC48 using agrobacterial mediated transformation. Examining of 92 transgenic plants revealed that 40 harbored at least one C2T substitution in the target region. Among the mutants, we obtained some single base substitution or 2. We also have 2 double base substitutions, and we also obtained a triple base substitution. The deamination window was spanning from 3 to 8. Then we also delivered this system base editor into wheat immature embryos by particle bombardment, and we regenerated the plants without herbicide selection. So we obtained 2 independent mutant plants. Sanger sequencing revealed that both mutant plants had C2T substitutions. One, we obtained the single or 3 C2T, and one obtained 1 C2T. And the PCR screening with 6 primer sites showed that both mutants did not carry the transgene vectors. So we noticed some limitations of the red APOBEC1 based CPE system. The genome target scope of BE3 is still restricted by narrow deamination window. Normally you see it's 5 base pairs, and it has a striking preference for TC and substantially decreased activity for GC context. So all this, and also at some target size, we observed very low efficiency of C2T substitution. So all these limitations affect the ability of a base editor to generate precise mutations. So then we need to develop a new base editor. Therefore, we screened more than 10 cytidine deaminases. They were mainly from APOBEC family member, and we identified a human APOBEC3A deaminase. This new base editor offers a larger editing window and a high efficiency of C2T. So you can see here, we applied this new base editor to target 20 different target sites of endogenous genes of wheat. The first one is wheat, rice, and potato. And the results showed that this new base editor had a very high C2T efficiency or substitution efficiency. They are, in general, they had, it gave 13-fold higher than the previously established red appleback-1 plant-based editor. So here you can see the red color, here is the efficiency, and the red color represents A3A, and the blue ones represent red appleback-1 based PBE, plant-based editor. And more importantly, it could efficiently target regions with TC dinucleotide independent of sequence contact. Here is an example. All these Cs, there's a G in front of all the Cs, and we could only observe A3A efficiency, but none of the PBE can work or can make this substitution at this GC content. So then we see, also we observe here, we observe A3A PBE edits cycling almost equally well, regardless of context. And then here I give you an example of using a plant-based editor to create some valuable traits. So we know weeds are major threat toward food production, and the herbicide-resistant varieties helping cost-effective tools for helping farmers manage weeds. So ALS here is a key enzyme in the biosynthesis of branched chain amino acid. It has been reported that amino acid substitutions in this enzyme would confer different herbicide resistance on plants. So as weed mutation at this ALS-P174 probably confer tolerance to herbicide. So we designed guide RNA targeting this region, and we got 4 different amino acid changes caused by the base editor, including P2S, P2A, P2F, and P2F and R2C. To determine whether these mutations confer herbicide resistance, so we transferred those mutant plants to medium supplemented with herbicide. So here is an example, and this mutant contained 5 edited alleles. You see, aging both have been substituted as the same in B genome, but in D genome, 1 allele is wild type. So we could see this mutant plant grew very well, whereas the wild type plant died. Here's wild type. Then we examined whether this mutant potentially could be used in the field for weed control. So here is the trans-T3 T2 plants harboring different number of edited alleles were exposed to herbicide at the field recommended rate. So we found mutants bearing, so here small letters is mutants. So mutants bearing 4 to 6 edited alleles could normally grow, whereas wild-type plants died within 30 days. Mutants with only 2 edited alleles showed growth retardation, suggesting that greater number of resistant alleles conferred potentially could be conferred a higher level of herbicide tolerance. So then this mutant with more edited alleles could be used for weed control in the field. We recently, or from last year, we also combined cytidine deaminase with adenosine deaminase to obtain a fusion deaminase. We named this system as STAM, standing for saturated targeting endogenous mutagenesis editors. In this system, a single protein using a single guide RNA would perform C to T and A to G simultaneously, and this STEM system acts as a dual base editor. To test the ability of the dual base editor to achieve saturated de novo mutagenesis in rice, we targeted the rice ACC gene. So ACC is an enzyme in lipid biosynthesis, and its CT domain is a target of herbicide. So here we designed 200 single guide RNA to target 400 amino acids of the CT domain. So we performed transformation, and the 200 guide RNAs were divided into 27 groups. To identify the designed mutants, we sprayed a commonly used ACC inhibitor to select for herbicide-resistant seedlings in group 1 to 27. So before spraying herbicide, we did NGS analysis. So here you can see from these two figures, you can see 92% of the guide RNAs have been successfully introduced into the plants, and 209 amino acids out of 400 have been changed. 3 weeks after spraying herbicide, several normal-looking seedlings appeared and were clearly herbicide resistant. Sanger sequencing showed that 10 in group 6, those 10 plants in group 6, carried the mutation 1927 from P to F. And the 2 seedlings in group 20 had the mutation W2125C. So the dual base editor could be used for directed evolution of most protein coding regions in plants. Although on-target conversion by base editing are found in many organisms, their off-target effects have not been systematically assessed at the whole genome level. So we performed a comprehensive investigation of the off-target mutation of BE3, HF1BE3, and the ABE by whole genome sequencing in rice. WGS data show that the number of C2T SNVs in the CBE treated groups was significantly higher than those reviewed by control and the ABE group. So this C2T SNVs associated with CBE occur more frequently in transcript genetic region where single-stranded DNA is generated owing to active transcription. So we concluded that cytosine-based editors induce genome-wide off-target mutation. So to increase CBE specificity, we first established NSA-Cas9 mediated R-loop assay, and in this assay, SA-Cas9 nickase was used to create single-stranded DNA region in plant cells that acted as a target for single guide RNA independent deamination changes. So using this transient system, we assess the specificity of 25 rationally designed truncated ApoBank3B DNAs, and we identified two cytidine base editors, VHM and TGR, with high specificity. So finally, I will conclude my talk that base editors enable efficient direct conversion of a specific DNA nucleotide into another in plant genome. And the dual base editors we develop are effective to induce or to increase the mutation diversity at the target site using only one guide RNA. And our WGS data showed that cytosine, but not adenine base editors induce genome-wide off-target mutations. And we established a very high throughput NSA-Cas9 mediated R-loop assay that enables specificity analysis of base editors in plants. We also obtained the two base edit or CBEs of VHM and TTR with high specificity and precision in plants. So finally, I would like to thank my group for their hard work and my collaborators for their very good collaboration. And the fundings are from National Natural Science Foundation of China, Chinese Academy of Sciences, Ministry of Agriculture of China, and the Ministry of Science and Technology. Thank you so much for your listening.
Again, another very valuable presentation. I had the pleasure of visiting Professor Guo's lab in Beijing, and I know she has an extremely extended and impressive setup. So we need to end our collaboration. Okay, Sultan, let's invite the third speaker.
Okay, sir. Thank you. Dr. Aftab, are you there? Can you introduce?
Okay, sir. Thank you so much. Our next speaker is Professor Yinong Yang from Penn State University. And Professor Yinong Yang is our collaborator from 2015 in our project of Cas as well. So Professor Yinong Yang has developed one of the earlier tDNA based CRISPR-Cas system for multiplex genome editing. And he has also been working in base editing as well. He has been working in plant pathology and working with rice and mushrooms as well. He has very excellent papers recently as well. So ladies and gentlemen, over to Professor Yinong Yang. Thank you so much, Professor Yinong Yang.
Well, thank you very much for the introduction and invitation. Yeah. Sorry, can you hear me?
Yes,
we can hear.
Okay.
You're very clear. Very clear.
Yeah, it's my great pleasure to give a presentation to the University of Agriculture in Pakistan. And so my today's talk will focus on use of CRISPR-Cas genome editing for precision breeding and disease management. So my research is mainly focused on rice disease resistance for the last 25 years. So we mainly work on mainly work on rice blast disease. We also are doing some work related to sheath blight in a few years ago. So usually when we try to understand the disease mechanism and also extend that to translate that to biotechnology, typically we're using a transgenic approach. For example, in this case, we overexpress a gene called ACC synthase, and this is under the control of a promoter called PB10, PR10 promoter, and that's kind of inducible only when that pathogen attack or induced expression of this ACC synthase leads to increased level of ethylene at local level and result in a disease resistance. However, these are transgenic plants. So there, as you know, there are a lot of limitations with a transgenic plant. One thing is that if you're using agrobacteria that mediate transformation or particle bombardment, and typically you got a random insertion of transgenes, these are not suitable for targeted mutagenesis or targeted insertion. It's very difficult to perform gene replacement to create any kind of allelic variation, as mentioned by previous two speakers. And that's also results in the introduction of undesirable DNA, such as tDNA and antibiotic selection marker. So that leads to very extensive government regulations requirement for GMOs, and obviously public also have concern regarding the genetic modified crops. So a new technology is much needed to precise and efficiently edit the genome for crop improvement. And hopefully this approach will also help to alleviate the public concern for genetically modified crops. Since the initial funding of that CRISPR technology, use of CRISPR technology for gene editing, our lab have started to adapt this tool for plant genome editing. As you see from this slide, CRISPR Cas9 conceptually is pretty simple, only require two components. So one is the Cas9 or other Cas protein, let's say Cas12 or Cas13. And the another component is the guide RNA. So guide RNA typically in case of Cas9 is a fusion of CRISPR RNA and transit RNA. So you have this guide RNA spacer and this guide RNA scaffold, which is a trans RNAs. So these two components with that guide RNA direct Cas9 to a specific genomic site. And in case of Cas9, they usually cut exactly cut three base pair upstream of PAN. So that result in the double strand break. And the subsequent repairments leads to either indel, or if you introduce a donor template, then you can perform that homolog dependent repair. Dr. Hogue already presented an excellent introduction regarding the homolog dependent repair. So I just briefly go through some of the basic strategies. Typically, you will design a specific and effective guide RNA spacer first, and then you will making a plasmid construct, or you can simply use the ribonuclear protein that not even involve any DNAs. So this construct or RNP complex can be introduced into the plant cell either by agrobacterial-mediated transformation or let's say particle bombardment or PEG-mediated transformation. So once you introduce the editing reagent into the plant cell, you will subsequently regenerate plantlet, then either using tissue culture, or sometimes even people do so-called implant genome editing. After you obtain that regenerate plant, you will do genotyping and molecular characterization, and followed by phenotyping and also functional analysis of particular genes, and you can evaluate the agronomic trait for the crop improvement. So this is kind of basic process that involve in that plant genome editing. I can just show quickly. So like several years ago, we have designed this website mainly allow you to perform guide RNA design. Nowadays, there are many of that website available, so you can design a specific guide and for you to making the construct. So once you design this guide RNA, and then you will clone into a particular type of the vector, whether it's a plasmid vector for maybe suitable for protoplast or for agrobacteria-mediated transformation. You can also just synthesize this guide. There are a number of companies that allow you to synthesize this guide. Then you can either complex with the protein or clone into the vector for genome editing. So for this example, you can introduce the plasmid DNA into protoplast, and then you can see that Cas9 is expressed, and then that will lead to the subsequent genome editing. So this is a transient assay we often do sometime before you perform the stable transformation. Obviously, for some of the crop, like especially Solanaceae plants, you can even regenerate the protoplast. Rice, you can regenerate plantlet from protoplast, but it's very difficult, and you also have a lot of somatic variation with regenerated plants. So in this case, we typically use agrobacteria-mediated transformation. Then you can regenerate plant. You can genotype them by restriction enzyme digest, or you follow by DNA sequencing. For example, you can see this particular gene is have a single base pair insertion or deletion, and sometimes multiple base pair insertion or deletion, which we call indel, and that leads to the knockout of a particular genes. So this is a target mutagenesis of single genes. In a lot of cases, actually, you require a use of a multiple guides to target multiple sites. Let's say Dr. Holger Puchter just mentioned about inversion and chromosome engineering via, let's say, inversion of translocation. So that requires multiple guides, and also for editing multiple bases, you maybe require multiple guide RNA for so-called multiplex editing. So typically, you using the multiple cassette. Let's say you have a polymerase III promoter, let's say U3 or U6, and guide a specific guide RNA. And then you have a terminator. Then you start with another promoters and guide RNA. But that it's kind of cumbersome to make a multiple guide RNA, whether from cloning standpoint view, or also if you have many of these, and then the construct will be very large. It's not very easy to manipulate with the binary vector, or especially if you're using a viral delivery, they're limited for the payload for the viral virus to deliver the genome editing reagent. So a few years ago, we developed a technology so-called polycistronic tRNA guide RNA technology. This is take advantage of the endogenous tRNA processing vehicle system. So in this case, basically you have a pre-tRNA, and we all know this pre-tRNA has to be processed to become a mature tRNA that involving the digestion and removal of 5' leader and 3' trailer by RNase. And so we take advantage of this particular processing system, and we arrange the tRNA alternate with this guard RNA and tRNA guard RNA. So we make this tRNA guard RNA array. Basically, you will lead to a transcription of a long tRNA guard RNA transcript. And that will be efficiently processed by endogenous tRNA processing enzymes and produce the multiple guard RNAs. There's a number of advantages with using this system. First is very precise, and it will cut exactly the 5' end at exactly location. 3' processing is less precise, maybe have extra nucleotide produced. However, this does not really affect the guard RNA activity, or that it performs its work. The other thing is that there is no obligation to start with a specific nucleotide, whether for U3 or U6 or any other post-3 promoter. Some of you maybe know that U3 promoter typically you want to start with A, and U6 promoter you typically want to start with a G. But in case if you're using the tRNA processing advantages, then there's no requirement to start with A or G. The other things, you can even use the polymerase II for that expression of guard RNA. So that's allow for more temporal or inducible expression, so temporal or spatial expression. The lastly, tRNA itself actually is an internal enhancer, and that will lead to the high expression of guard RNA, and that also result in some much high editing efficiency. So you can see from this example, we target that mark gene of phytoin desaturate synthase in rice. And we designed 2 guide target that particular genes. In the first generation, we're able to get 87%, and so close to 90% of the seedlings that edited biallelically. That means both chromosome copy are edited. If you just counting for the monoallelic, it's almost achieved 100% efficiency. Obviously, you target the 2 guide, that's just even make high editing efficiency. If 1 guide, it may not necessarily reach the 100%, but with the both guide, so you can reach a very high efficiency of genome editing. Obviously, this method is mainly for multiplex editing, so you can target, in this case, we target a genomic site, and they will all be edited simultaneously at a genomic site. And so some site, if they added the deletion occurred, editing occurs simultaneously, then you got a deletion fragment. So you can add this. We have tested up to about 10 guard RNA, but I believe you probably can go even high with the high number of guard RNA. So this is not only just for indel or introduce deletions. You can also use a similar approach for base editing. So in this case, we actually target 2 gene called rice white strip leaf and a zebra 3. These are editing of the single base pair change from A to G will lead to a specific phenotype. So we use this tRNA and also introduce two guide and to perform the base editing, so A to G. So base editing, Dr. Seisha Gao just gave an excellent presentation. She has a very sophisticated process. In this case, is a ABE editor, so it's a editing base editor, which provides usually a more precise, has less off-target issues. So you can see here with that first with the Z3 locus, we got very high efficiency. I mean, up to 39% editing efficiency in one of the base editor. Another was different version of ABE is less high frequency. However, when we with the BSL5, we got very low editing, in only 1 to like 3%. There have been other reports also demonstrate sometimes there are locus dependent. Also, it could be just simply because that WSL5 that for that particular guide RNA is not in that perfect editing windows. So you can see that after you editing from change from A to G, and you can result in the expected phenotype, which in this case zebra is the kind of zebra strip, and in this case is a white stripe, the leaf. So this approach is not, I mentioned, is can be used for multiplex. You create indel. You can also for base editing. In theory, you can also do homolog dependent repair. Obviously, this is extreme difficulty in plant system. However, in fungus, this actually is a more predominant. In many of fungus are also in bacteria. This actually is a more predominant repair pathway. So in this particular case, we use the rice blast fungi as an example. We introduced a 3 guide and added 3 site, and this editing a small gene family called NLP, which is an effect of fungal blast pathogen. And you can also see that we supply 3 donors, and then you're able to got a pretty high efficiency, especially for the NLP1 and NLP3. So we able to got a very high efficiency of homolog dependent repair with these 2 locus. We found NLP2 in fact was located nearby centromere for some reason, has a very low level of homolog dependent repair, but relative high level of NHEJ. Rice blast fungus still can have some NHEJ activity, but predominantly actually it was based on the HDR for DNA repair. So we were able to obtain single, double, and triple mutant using this approach. So you can also already see that this method is not only be used in plants, and it can also be used in microbe. There also we also did a human mammalian cell culture, and a number of the lab have test different kind of animals and plant and microbial species for the broad application of this particular approach. So obviously, CRISPR-Cas has a lot of application in basic science, in plant functional genomics, and as well as crop improvement. For functional discovery, you can editing any kinds of plant gene, whether it's regulatory, enzymatic, or structural gene, protein genes. You can also edit in the promoter, intron, and the small RNA or non-coding RNAs. You can even edit the repetitive sequence. People have been used that to remove some of the rich element. So genetically, you can perform a loss of function analysis. You can also do a gain of function analysis by knock-in or transcriptional activation. And if you're doing cell biology, you can use CRISPR fused with CRISPR-Cas fused with GFP, IFP for labeling and imaging process analysis. People also use CRISPR for epigenome editing. So that's kind of methylation or demethylation or even acetylation of a histone. So there are a lot of utility of CRISPR-Cas for crop functional genomics. So this is just a quick example. We have performed a couple of years ago a gene called the PTR, which is a broad spectrum resistant gene, non-typical. This is a non-NLR resistant gene that was originally map-based cloned by a USDA scientist. So they asked us to help to validate this gene is indeed rice blast resistant gene. So we simply perform a knockout of this gene, and you can see by knocking out that particular gene, and this is a hypersensitive response, this resistant response now become a susceptible response. Now you have a lesion formation in this susceptible. Originally it was a resistant line, now it becomes susceptible. So this is a simple use of technology to validate gene functions, and that leads to discovery of novel rice blast resistant gene. You can also use that for function discovery of even essential genes. So we have identified a couple of essential genes belong to the family of megakinase in rice. So we find at least two megakinase was involving, one was involved in the embryo development, another was involved in the female gametophyte development. So there obviously there are many other utility for functional discovery. I just give these two examples. Many of you are probably more interested on the crop breeding using this technology. And the first wave of that CRISPR editing in crop breeding is usually as a targeted mutagenesis, because it's much easier to create a knockout and then site-specific insertion or the gene replacement. So you can knock out, let's say, a negative regulator of the plant defense immunity. You can create a disease-resistant plant. You also can alter in the flower time and growth development by editing genes and promoter. There's a number of examples. So you could also editing some gene involving the browning of some of the agricultural product. A lot of research has already been done by modifying starch property like waxy mutant, and also oil composition and hybrid breedings like male sterility. So there are many work already been done, and some start to used in initial commercialization, but not quite, but it's the pre-commercialization stage in many of the products. Obviously, there are another two area is like a more precise genome editing, which is like site-specific integration of gene stacking, like you can stack multiple resistant gene or herbicide resistant together in the same chromosome locations. Probably the most profound application will be a precise genome editing, such as using the base editing, prime editing, or HDR to create allelic variation, because a lot of these allelic variation are responsible for high yield, quality, and disease resistance and abiotic stress tolerance. So I just give you a couple examples we have done recently. One of the example is the editing of a gene called IPA, ideal plant architecture. So this gene has been characterized extensively, particularly by a couple lab in China, and demonstrate these genes involving potentially involving heterosis and also involving yield improvement. So interestingly, this gene has to be like control at a precise level. So this gene, the expression, if it's too high or too low, it will not give a high yield, but its optimal expression level, it will result in a high yield. So we perform the targeted mutagenesis of the IPA promoter. So we position 8 guide along the promoter region. There are a couple guide actually particularly focus on the region was GC rich. People think that was the region responsible for methylation and suppression of that gene. So if you delete, let's say, this particular methylate, that the GC rich region, it may be increased the expression because of a relief of the epigenome, the methylation. So we generate over 180 mutant line, and we basically just screen this line for high yield and like some of these the trait associated with the IPA. Significantly, we perform this genome editing in elite commercial line called Jupiter, and this is a predominant medium grain rice grow in southern United States. So we thought by editing this particular cultivar, we may be able to quickly translate this for potential commercialization in the near future. So this slide shows that in that, I think this was T1 generation, and you can see that the editing of the promoter region will actually result in a variation in the heading date. So this line actually, we have a lot of those line actually is transgene free after the 1 generation, the segregation of the Cas9 construct. So that's another important, because if it's transgene free, USDA will not regulate this for as a genetically modified the crop. So you can see some of this line actually has much early flowering. Obviously, some delayed flowering. I was told by a breeder, they actually was interesting to obtain some of the line like 1 week early maturation, so they can plant some other crop subsequently. So you can see by altering the expression of this particular gene, you can change the that flowering time. And also it will impact the grain weight and panicle number that potentially leads to a high yield. So this dashed line is basically is a control of yield of control plant, white type plant. And you have see some of those lines actually have a high total grain weight and panicle numbers. Obviously, there are a lot of them actually is getting worse. It's a low level of that grain weight. And so so that's for the IPA. Another, we also did editing of number of the rice blast gene editing. And in this particular case is a gene called a PID3. So this is a rice blast gene confer resistant to particular races carrying the AVR PID3. And however, in the elite line, Jupiter. So this Jupiter and also Nubberberry, which is a type strain for the sequence strain. In both cases, actually don't carry the functional IPD3. So these two are the resistant line and is IPD3 resistance. And they have a functional or complete NLR resistant gene. However, in these two cultivar, include this allele cultivar, there is a stop codon here. And by using adenine-based editor, we can convert stop codon to glutamine, so that restore the functional the allele. So you can see that so A to G. So this basically is that T to C is the same. So we're able to restore that functional allele. And this also can be evident by the enzyme digestion, because that conversion also happens to create the BAN-H1 restriction site. So you can see now that this is still a monoallelic. Recently, we already got the biallelic editing line by Selfing, and characterized homozygous biallelic lines. So we hopefully to test this resistant line, also that the IPA, the yield, high yield plant in the field. However, this year, due to a pandemic, we're not able to perform any field test. But hopefully in the near future, we're able to perform this in the southern United States for field evaluation. However, there's still a lot of concern, public concern of GMO in the United States. Nowadays, for the gene edited like a plant, the rice plant we mentioned, this will not be regulated, because if it's just simple deletion or single base pair mutation, they're not subject to regulation. And that was the example set using our early work with the mushroom, knock out a gene called the PPO, polyphenol oxidase. And by knocking out that gene, we try to reduce the browning. So they use that example to set that basically the guideline for regulate that CRISPR edited plant. And also DuPont has a Waxicon. I mentioned about Waxicon are basically those has a high amylose activity. So those are sticky starch corn. However, both of these has not been commercialized though. And that's the other reason, even when we try to perform rice field tests in southern United States, there are hardly many of the breeders interesting, because they are kind of afraid with some of the potential implication with that genetic modified rice, because in United States, rice export to European Union, Japan, and Middle East. So as you know, Dr. Holger Puchter just mentioned, the EU has a law that basically kind of almost like a ban on CRISPR-Cas for commercialization. Well, they basically treat this as a genetic modified crop, same as the early generation of transgenic. So because all the trade are interlinked. I mean, so the trade is not just limited in the United States. It's actually is a global trade. So if you have CRISPR-Cas edited crop in, let's say, in Arkansas, in southern United States, and you are landed in European country, and that they don't allow the commercialization of CRISPR edited crop, then you're in trouble, you're in problem. So since that, in fact, the only story of that the CRISPR mushroom, I sometimes got these emails that this postcard from that Canada, where people kind of afraid of the frankenfood or whatever. So we really need a more rational dialogue, and also a more coordinated regulatory frame across different nations. Finally, I just want to give an example of that's probably the less controversial, and so this is actually use of CRISPR for disease diagnostics. So in this particular case, we try to detect a bacteria called Candidus Leprobacter asiaticus. Many of you know that this causes a major disease called citrus greening, also called Huanglongbing. This disease was devastating to the Florida citrus industry, wipe out up to 75 to 80% of their citrus production during the last 10 years. And this disease is usually is very low titer, so that means present is very low level, and it's not easy to detect. It can be done with real-time PCR, but sometimes it's also not so easy to detect. Now we know that there are a number of technologies have been developed using CRISPR-Cas to detect that DNAs and RNA at a very high sensitivity. So in this particular case, we're using the approach developed by Jennifer Doudna's lab, and this is using Cas12a. Feng Zhang also have a Cas13a detection, and also both Cas12 and Cas13 detection. So in this particular case, you will run so-called recombinase polymerase amplification first, and this is isomer amplification, so it's 39 degrees. You can just use a heat pad. So this is an instrument-free detection method. You don't really need a PCR machine or anything. You basically heat at 39 degrees and you have RPA. Then you follow by Cas12a detection. So this allows very high sensitive detection of this particular bacteria. So we can reach automolar detection level. So this with the detector. You can also detect with Cas12a alone, but it's probably 1,000-fold less sensitive. So you can see we test different samples from Florida. So sweet orange, pomelos, periwinkle with alternative host, and also citrus psylla, which is an insect that transmits disease. So we can readily detect those infected samples. Also, you can adapt this with a simple lateral flow assay. So you can see this was able to detect at 1 automolar level. And so these are some of these samples. Recently, we also improved this method by further optimize some of the primers and guide RNAs, and that will even give a high sensitivity and low background. So hopefully these are less controversial. However, in our lab, we are kind of more focused on recently on the more precise genome editing, such as indel prediction and base editing, and Dr. Teisha Gao mentioned, and also another technique called prime editing. I know Teisha and probably Hoge, you also work on that. And then we also try to make some improvement on the HDR, but we haven't able to make a lot of progress. And this technology can be used for different kinds of genome editing, whether it's multiplex, some could be DNA-free. And hopefully we can improve the crop productivity and quality, and by, let's say, modify the heterosis, and that maybe increase high yield and also more superior quality. Obviously, this is for, that's important for food security. The other thing is if we develop a disease-resistant or insect-resistant cultivar, and let's say increase use of nitrate use efficiency and drought tolerance, that in fact reduce the pesticide fertilizer usage, and this could really improve environment. And so it's this in fact is compatible with organic farming, because even for organic farming in the United States, people still use to a certain level, so-called certified approved pesticide. So they're still using some level of pesticide. So with the genetic approach to improve the crop disease resistant abiotic stress tolerance, that really work well with the means a green agriculture concept. So eventually we hope to use genome editing for a more sustainable agriculture and environment. So finally, I would acknowledge some of people who have been involved in this work, particularly Ka-Bin Xia, who spearhead this research, and Bastian, who recently just started a new job in a company after postdoc with Jennifer Doudna and Brian Stasiewicz. Katu did the base editing. He is currently in India. Matthew performed that the disease diagnostic work, and Justin is mainly work on HDR and prime editing. So our findings mainly from the United States Department of Agriculture and National Science Foundation. So we really don't have rice plant in Pennsylvania. We grow this in the greenhouse in our fourth floor greenhouse. However, we do work together with people in the southern United States for some of our rice research, particularly the field test in the near future. Well, thank you very much for your attention, and I will be happy to answer any questions.
Okay, Dr. Sultan, I suppose we are at Q&A level now.
Yes, sir.
Okay, you know, I looked through this Q&A chat box.
And cotton and wheat. If you have expertise in this field, we would highly appreciate if we can have some training on it. Right.
So-
My question is for both of you.
Okay, so I'm unfortunately I'm not able to give you any suggestions because we are not working on this, but it might be different for Yin and Yang.
Well, obviously, I mostly work on the rice, but tomato, are you mentioning about tomato? Did you?
Yeah, I'm specifically asking for nucleoprotein transfection system. We are transforming
IMP, is that nuclear?
Yeah, protein system. So I mean, we are transforming Cas9 and plants are transgenic, then we have to segregate them. So the straightforward system is transfected nucleoprotein. So if you have some expertise, if we can have some training on it, that will be perfect.
Yeah, so my lab don't have a whole lot of experience with work with IMP, except we did some work in fungi. I mean, obviously that's much easier to do that with the IMP because you can regenerate fungal cell relatively easily. We did some early work with potato, but that was with the plasmid. However, there are a number of labs, well, particularly early work done in South Korea. They have done with IMP, but those cases usually regenerate protoplast out of that like lettuce cell. This usually is more challenging. There are a number of labs working on using maybe using, let's say, a particular delivery approach, let's say some of narrow material or things like that. So you basically either you go by a protoplast regeneration approach, or you use some sort of implant editing. I mean, using probably the narrow material.
Right, right. So I think there will be some.
There are some article already published, but implant editing still is kind of challenging. Dan Voigt's lab has using virus, obviously recently. He has a couple of paper published. He was using TRV and also using meristem induce the, let's say, some baby boom or washout gene to facilitate the regeneration of the shoots in planta.
Right, right. So yeah, thank you very much. Yeah.
May I also make a short comment to this? So in principle, it was very interesting for European in the beginning to use RMPs because everybody hoped that this way we could escape the ruling because the RMP is a chemical and not a DNA molecule. And so I also know from a lot of groups that had quite difficulties to obtain high frequencies when they applied RMPs to different plant species. I think that Gao indeed was very efficient, was reported on high efficiencies with these technologies. And maybe you ask her if it comes to how these things are done best.
Yes,
Yeah,
thank
so-
you.
So I also want to comment on this that yes, Sasha Gawa has been working on RNPs and she has developed the system and has made some mutations in wheat as well. So when I visited her lab in 2016, so they were she were developing this system. So she is very good in this regard. So my question is that when we go for the DNA-free transgenic plants, especially to avoid the regulatory issues, although in 2015, US regulatory authorities, they allowed some very precise small deletions to not be regulated. So in this regard, apart from RNPs, when we are going to make DNA-free plants, then what would be the better choice? Like we have to go for the segregating populations, or there are other choices to create DNA-free plants to avoid regulatory issues?
So if I'm allowed to make a short comment, so in principle, I think that at least according to the ruling in the European Union at the moment, it doesn't matter whether you use RMPs or you use a transgene, it's all the same. But from our experience working with transgenes, depending of course on the plant species you use and segregating it out afterwards might be more efficient because the mutation rates are much higher. As we heard before, if you are not able to really get mutations by using RMPs, it doesn't help you so much. So in principle, it's always a matter of efficiency, I guess, how efficient you can get with this method, whether this is an alternative you really want to take or whether this is more burden for you. But in principle, I think all regulation, as far as I see it, if it comes to segregating out the transgene, and it's clearly shown that it's segregating out, it should be GMO-free, regarded as GMO-free, yes, in many countries.
Yeah, in the United States, it doesn't really matter if you use RNP or using the DNA construct. I mean, if you create deletions or if you introduce the DNA from relative species, that those are sexually compatible species, then they are not subject to regulations.
Right, right. Thank you. Any other question? Is there any other question?
Well, if there's no more question, we know that our honorable speakers are accessible via email and through other social media channels. We remain indebted to their time and effort that they put into enlightening us this evening. On behalf of all participants and on my personal behalf, we are extremely grateful and we look forward to continuing our interaction in future. Thank you so much.
Thank you so much for inviting us.
Thank you very much.
Thank you. Thank you, sir.
Yeah, thank you so much, Professor Holger, as well as Professor Yinong and Professor Sasha Gao for their time from very busy schedule. I can understand you have very busy schedule. I'm so grateful to our respected Vice Chancellor as well that he spared time from his very busy schedule as well and permitted us to arrange this event and asked us to contact you. Thank you so much. Thank you so much.
Thank you.
Thank you. Bye-bye.
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 Thank you. Bye.